Over the next 45 days, we've been exploring the complexity of the mind with neuroscientists, psychologists, philosophers, artists, scholars, and more. And today, as a part of our public lecture series, we're very pleased to welcome Professor Shubha Tole. And today she will be delivering her lecture titled Sensory Perception, From Genes to Brain Circuits. So I've had the pleasure to know Shubha from my master's day, so I'm very delighted to be able to introduce her to you all today. Shubha is a neuroscientist at the Tata Institute of Fundamental Research in Mumbai. Her seminal contributions to understanding how the brain develops in an early embryo have been recognized by prestigious awards such as the Shanti Swarup Bhatnagar Award and the Infosys Prize. Shubha leverages her experience as a mentor, policy maker, and a senior scientist to actively engage in science outreach that inspires younger scientists and is a vocal advocate for women in science and mental health awareness in academia. Before I hand over to Shubha for today's lecture, I'd also like to mention to all our participants today about some of the programs that are coming up next weekend. Do watch our film, Eyes of Stone, which is currently being screened in the theatre section of Psyche, and join us for a discussion between filmmaker Nilita Vachani and psychologist Purnima Bhola on 21st April at 6:30 p.m. in the evening. And if you enjoyed the 3 episodes of Hamlet's Live that happened this weekend, do tune in for the 4th episode that's happening next Friday on 22 April at 5 p.m. I'd also like to remind the audience that there will be a Q&A session at the end of the lecture, so do type in your questions in the Q&A box and do share your feedback with us. So there's a link for the feedback form that will be dropped in the chat, and it's always helpful for us to know what we thought about the lecture and what we can do better in our public programming going forward. Now, I'd like to hand over to Shubha to start the session. Thank you so much.
Thank you so very much, Madhu. It's very nice to be invited to this wonderful series. I think you folks have come up with a very creative angle to engage the public and to start conversations, which is really wonderful. And of course, I have very, very happy memories of when you were a master's student at TIFR, and it's great to see you doing such cool, creative things. So let me begin to share some of the excitement of the brain and of neuroscience that I have had the luxury of studying, and a cool discovery that we had the privilege of making in my lab at the Tata Institute. And I hope you will all find it an interesting story. There. Okay, cool. So this is a glorious multicolor view of the human brain. This color is actually, it's color coded for all of the wires of the brain, all of the nerve tracts. So you can see this entire cable, as it were, turning over like this, another cable bending over in a U, another going forward. The colors are actually artificially colored for every time the cable changes direction. So that's the kind of imaging produced by the Human Connectome project. And I wanted to share this to bring out what complicated circuitry it takes for our brains to work. Our brains do fantastic things. Our brains allow us to perceive what's going on in the world. We take in that information from our 5 senses, we process it, we create an impression, an image of what's going on outside so that we may move through this world. As we move through this world, we have a good sense of where our body is and how we may fit into, let's say, a small airplane seat, an expansive sofa. We take information about the world so that we may live in it and so that we may navigate it. It's safe to say that everything that we know about the world comes through us to our senses. If we didn't have our senses, we would not know the world. And then it again is a corollary of this, that all we know about the world comes through our senses. All we know about the world. There may be magic going on outside, but if we cannot sense it, we do not know. And I'm going to challenge this idea of our sense of how well we know the world. We humans are so proud about being, we're, so clever. We can control our world and we can modify, well, and also destroy our environment. Nothing to be proud of there. But we're so full of how much we can do. And yet, I'm going to bring out some of the limitations in our so-called wonderful senses. Is seeing believing? We trust our eyes. Aankho dekha hal. We trust what we see. But now, try this. What color are these flowers? I guess people, this is not a trick question. I see them as purple on the outside of the petals and then white on the inside with this yellow. I see this one as a yellow flower. Okay, this is from the library of Bjorn Roslet, who specializes in photographing things in ultraviolet light. We don't see in ultraviolet light. Okay, but if a photo is taken in ultraviolet light and then false colored so that we may see it, here is how the flower might appear to say an insect that actually can see in ultraviolet light. Okay, so the colors are false colored here, but the point is that within this, what appears to be plain yellow, there are differences in the way the structures of the petals reflect light, reflect ultraviolet light, which if you could sense ultraviolet light, you would be able to pick it up. And certainly you can see how the flowers now colored this way are probably more relevant to an insect who wants to find the center of the flower. So you can see this kind of bullseye phenomenon, right, which might help the insect find the center of the flower. So, you know, what color is the flower then? What color is the flower? Isn't it really based on who's looking? So is there such a thing as yellow color? It merely is, you know, what wavelength of light our paltry little visual spectrum sensing eyes were able to see, right? There is no color, it's only what you can see. And this is the theme I'm going to bring out, the limitations of our senses upon which we rely so completely and upon which we base our confidence that we know the world. What color are these dots? Okay, I guess people would agree, and I too, I see this as an orangish rectangle with a brown dot, and here a brown rectangle with an orangish dot. Excuse me. So this is actually just a little bit of a trick, because I have taken this very color and I have copied it and copied it and copied it and copied it. And lo and behold, I'm trying to tell you that each of these circles is actually the same color. I did not change things when I copied them. Okay. And since I didn't change anything when I copied it, but even then I can kind of feel that this browner on this side and redder on this side, right? It's our eyes playing tricks on us. And our eyes play tricks on us because of the limitations of our biology. So we're going to see it this way because our biology, our wires, our circuitry is wired up like this. And this tells you that context is everything. Okay. I'm going to give you another example of whether we hear with our ears, or whether our senses sort of collaborate with each other in what we're going to perceive. Okay, I'm going to play this clip and I'm going to just ask you what you hear. Okay.
Ba.
Sorry, it's frozen.
Ba, 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 ba.
All right. If this were an interactive session, I would ask the audience what they heard, and I'm pretty sure you basically ba ba ba.
Ba.
It's like baby
ba,
sounds, right?
Ba, 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 ba. Ba,
now,
ba.
it's going to knock your socks off when I tell you that these two audio tracks were the same. The audio track was exactly the same, and it was simply that the visual was different. The lips were making pa, pa, pa in the one case and pa, pa, pa in the one case, but the audio was the same. And here is the two videos put together. And now you can see for yourself. You can close your eyes, or you can hop from one to the other, and you can see what your brain is, or you can hear what your brain is telling you.
Ba, ba, ba.
does it freeze?
Ba, 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 ba.
Would you like to hear it again and try?
Ba, 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 ba.
So I hope you saw that our eyes affect what we hear. Okay, so if seeing is believing, then our eyes can control even what we hear. So this tells us that our circuitry is our circuitry is what we have to sense the world. Our circuitry is what we have to grow so that it wires up correctly. Our circuitry is really that which gives us the ability to make sense of what's out there. And now let's test the circuitry a little. Let's just move through this. Let's do a little thought experiment. Imagine a tennis net in front of you, and I'm going to ask you to close your eyes and poke the net and simply answer, does this net have holes? Does this material have holes? And of course, you're going to be able to put your finger or maybe your whole fist through it. And of course, you tell me it has holes, right? But now if I ask you to do the same test, does this material have holes with a mosquito net, you're not going to be able to feel the holes. You don't have the resolution, right? Now, if I increase your resolution by giving you a toothpick or a needle, you would then be able to ascertain whether the mosquito net has holes. So the point is, we live through our world so happy and confident with what we know, but our information is constrained by the resolution. There are things we cannot see. There are things we cannot feel. Indeed, even when we hear something, our eyes tell us something different, and that's what we believe. And in this kind of space is how we navigate our world. We make the best possible interpretation with very limited information. And therefore, how the nerves carry the sensory information to the brain and how the circuit gets wired up becomes an important question in biology. Because indeed, how this brain is built and how the circuitry is wired up, I wish indeed that one could plant an idea in one's head like that, but not so. So how the circuitry is wired up is really the name of the game, because if it doesn't get wired up correctly, the senses aren't going to function, and how we perceive the world is going to be affected. So here, take a look at these videos. This is on the right, a fish embryo growing, and you will see how in the space of a few days, it goes from egg, single cell, to a bunch of cells, and then actually a whole fish. And here on the side, a human, this is a cartoon, not the real thing. One cell becomes two, ball of cells, and this moves to the lining of the uterus, where it embeds in self and then grows placenta. And this blue here is going to become the embryo, this blue line. While here the fish is doing its thing, very much the same thing actually. And you can see the beginnings of what would be the vertebral column coming up there, the eye. And here, look, the same thing's happening with the fish. Look, the beginnings of the vertebral column coming up. Here the eye, okay. Here the limbs are beginning to form while the fish is forming its baby fins. And these two glorious creatures developing, right? Lovely tail elongates. This is sort of the magic of life happening. And even while all this is happening, the wires in the nervous system are growing and making their connections so that these little creatures will be able to navigate the world around them. Kind of amazing, right? Here in the brain then, look at how the brain cells migrate, okay? Everybody moves. This is like traffic in a city where each car knows where it's going, but my God, the overall view is so confusing. This is from a friend and colleague, Kazunori Nakajima, who runs a research lab in Japan, and he was kind enough to allow me to show this. Look here. Each cell is darting, rushing to where it wants to go so that the brain may assemble properly. And it looks confusing, but at the end of this one gets a functioning brain circuit. And this is what we developmental neurobiologists study. How does this fantastic circuitry get assembled in development? Now, here is a picture of a human brain, and here is a simplified map of the visual sensory pathway. Okay, and to give you a sense of how complicated it is, okay, neurons from the retina connect to okay, but here's a sense of the complexity. Here is one brain neuron, and every little red dot represents where some other neuron has come and connected to it. Okay, now each neuron makes between 1,000 and 10,000 connections with some other neuron. And the brain has 100 billion such neurons. So this is crazy, right? So this is where this magical and confounding riot of colors comes from. Now we study these questions that we want to study in a simpler model. We study it in the model system of the mouse. Okay, so here is actually real data from my lab. This is a mouse embryo brain, and you can see that this green bundle here is a bundle of wires that emerged from here, crossed over, took this nice U-shaped turn, and this is the bundle of wires that carries sensory information into the cerebral cortex, which is the processing center of the brain. This is how the mouse sees and how it hears and how it feels. And indeed, this pathway is what we have in our brains that control how we see and hear and feel. So you can imagine that this pathway's growth into the cerebral cortex is going to be critical in order for us to see and hear and feel. This is what we study. I'm going to show you a little example of the kind of data that we get. Okay, now, in order to be able to sense anything at high resolution, you need more and more, let's say, pixels devoted to it, right? I mean, you can imagine if you have a camera of low megapixels versus high, the higher one's going to get better resolution because the information is given to more pixels, right? Similarly, when the pathway takes sensory information into the brain has limited processing power, but it does a very clever trick. It doesn't uniformly take all the information and just use it equally. It's not an equal opportunity process. Some areas, say some areas of our body, if you're talking about touch, some areas of your body are more important in terms of sensation than others, right? Your fingers are more, you need to touch, you need to feel what you're eating and so on. So fingers or the lips, because you're going to eat things, some parts of our body are more important for sensation. And those parts get access to more territory in the brain. And that's how the brain achieves high resolution of, say, touch in some parts of our body. So if we drew a human being, okay, proportional to how much of the brain it had in terms of its sensory nerves, here's what you would get. Okay, here is disproportionate amount of brain power given to sensing touch on the face, specifically on the lips and mouth, because this is seriously important for life. No? Anybody who could not sense whether they were eating something horrible and pokey that was going to pierce their guts would, have taken themselves out of the evolutionary game. Likewise, the hands. And a similar thing happens for all species. Okay, so this is the normal mouse, normal shaped and proportion. And here is the same mouse drawn in proportion to the brain area that processes touch. Okay, the mouse's whiskers are like fingers. So before a mouse goes through a crack, it actually actively whisks to make sure the place is big enough for it to go through. And likewise, again, look, forelimbs, more important, hind limbs, not so much for touch. And likewise, upper jaw, lower jaw, and so on. Now these whiskers, okay, each whisker is like a finger. Each whisker touches a unique part of the brain, and you actually get this lovely little, map of ovals in the brain corresponding to each whisker. And that's how the mouse can sense. Like, you can tell if this finger is touched over this finger, right? How can you tell it's different? You can tell it's different because the nerves carrying touch from this go to two different parts of the brain. That's how you can tell whether it's this or that finger. Similarly, the mouse can tell which whisker has been touched, and that gives it resolution. All right. So that's what we study. Okay, this is the sensory pathway in cartoon that reaches the brain, that carries this information, in this case of touch. Okay, this is the same slide I showed you before. This is work of my former PhD student, Suranjana Pal, who produced this beautiful finding that got published in the Journal of Neuroscience. And here's what we asked. She asked, how does this circuitry of sensation form? And I'm going to show you one data slide, just to give you a sense. Now, what Suranjana did is that she examined a mouse in which a particular master control molecule was knocked out. The gene for it was mutated. Okay? And what do I mean by knocked out? If a gene is made inactive or broken and unable to work, then that gene does not make the protein that's supposed to come using the instructions coded in that gene. And then that protein can't work, or it can't be made. And then the brain has to somehow get along without it. So the consequences of what happens is what we examine. And that's how we draw ideas from, or hypotheses from, what that gene might have been doing in the first place in the normal brain. Okay, so I'm going to show you one slide of what happens when a particular master control molecule that Suranjana was studying was mutated in the brain. Okay, so here's a mouse. And here is a section now of the normal mouse brain. And remember, I said each whisker is connected to a little oval patch for itself in the brain. So these patches are literally corresponding to innervation from whiskers. And this is how the mouse can tell which whisker has been touched. And here it is in high mag. It just looks glorious. You can see these individual, these are single nerves. Okay, they have all grown because they knew where and how to grow. Well, now I'm going to show you a picture in which one gene, one control molecule is mutated. And when that happens, okay, when that molecule is gene is knocked out, basically the pathway enters all the way up to the cerebral cortex here, but then it can't make inroads in. It can't dive in and actually make these lovely oval patch connections, which means then that when this particular gene is knocked out in the brain, okay, while the pathway is fine, the mouse's eyes are fine, the ears are fine, the skin sensation the sensory nerves come from the skin, all these external organs are fine, but the mouse can neither see nor hear nor feel because these nerves are unable to connect to the cerebral cortex. So the signal cannot be processed. So this was her finding. And of course, we're taking it further to ask, hey, well, what's the problem? You know, what is the problem without this one gene that these nerves can't make the connections? And there we then get an understanding of how the brain is normally connected. Now, this was sensory perception. I'm going to now share with you a small vignette of how neurons are created in the first place and how the brain decides how many neurons is enough. Okay, neurons can't work by themselves. One would imagine the more the neurons, the more the processing part, but no, they can't work by themselves. They need support cells called glia. And in fact, without these support cells, the neurons would absolutely not be able to work. So the numbers of neurons and glia in the brain have to be matched. And we discovered a factor that actually controls this process. So what happens? Okay, what happens when we're now looking at a standard mouse cage in which mice are living, they have food, they have water. Okay, now we're going to look at what happens to their neurons when they're in a standard cage versus when they're in a cage that's called an enriched environment. Okay, enriched means, look, they have toys to play with, they have wheel to run in, they have little things to crawl through and so on. So this is an enriched cage. Now, in the learning and memory center of the brain called the hippocampus, the normal standard house mice have a certain number of neurons, and each neuron has an arbor like a tree. These are all the input wires, these are all the output wire. Okay. This is the standard mouse. Now, in the same part of the brain, an enriched environment mouse will have more neurons, and each of them will have more branches. So you can see the stimulated mouse is making more and more connections and actually creating more neurons. Now, we asked, how are neurons created and how their number is decided? Okay, so what we found is that brain stem cells, they normally produce neurons when a certain gene that we're looking at, a master control molecule, is on, and you get a neuron. Now, we found that when this gene is turned off, okay, it's knocked out, it's mutated, then you get the support cell, this glial cell. Okay? And then, amazingly, if we forcibly kept this gene on, we didn't get support cells, we only got more and more neurons. So we were able to conclude that this particular gene we're looking at, in fact, makes brain stem cells exclusively make neurons at the expense of the support cells. This is not a good thing, but this tells us then that the activity of this particular molecule we're looking at, in fact, normally controls which cell type is being made. Okay. So this is just an example of what we mean when we say that we keep this gene permanently on, we get more neurons. This is a normal mouse brain, okay? And here is a mouse brain with many. Each green neuron is a neuron in which this particular gene was forcibly turned on, and these neurons should never have been there. They should have all been support cells. So this is the work of a postdoctoral fellow in my lab, Dr. Archana Iyer, who's produced these beautiful images, just to show you how we are able to understand how neurogenesis and gliogenesis, support cell making, is controlled in the mouse brain. So I will just wrap up this my talk now by simply summarizing how in this riotous, colorful map of the human brain, we achieve insights using the mouse. In the mouse, we've discussed how the sensory pathway that brings sensation into the cerebral cortex actually depends on a particular molecule that we're looking at, which, if it's mutated, this pathway can reach all the way up to the brain, but it can't enter and make connections. And then this mouse can neither see nor hear nor feel. So this is a master control molecule, and that's sort of one insight that I've shared with you. And another insight is that when the stem cell is making neurons in the first place, okay, it has to decide when to stop making neurons and when to switch to support cells. And to do this, a particular control molecule is required. And if that control molecule doesn't go off and continuously stays on, then you get only neurons and you don't get any support cells, which is not a very good thing. So I've shared with you two insights about the development of the brain and the motivation to study the development of the brain also, which is that if you don't know how a circuit is normally assembled, you're not going to be able to understand what happens when things go wrong. Okay, why are disorders such as autism, schizophrenia, mental retardation, cerebral palsy, what goes wrong in these disorders? Unless you understand the normal framework, these disorders will not be tractable. So studying how this masterful computer is assembled is the sort of research interest and passion of our lab. And I'm delighted to have been able to share some of that with you today. Thank you.
No worries. So there are a couple of questions in the chat, and I can start with that, and then we have some more time to go. I think the first question that came in during the talk while you were speaking itself was from Bhavi, and they wanted to know how I guess this resolution would be achieved, but maybe rather what happens to this resolution in the case of amputation or the loss of limbs, or maybe the loss of any kind of sensory input organ.
Oh, wonderful. Okay. When a limb is amputated, the sensory nerves from that limb, or rather, so the sensory nerves that take sensation from that limb, the neurons actually reside right next to the spinal cord. So those neurons are still there, and their input wires have gone all the way to the limb, but those input wires are now cut. So now there's nothing for them to draw input from. Now, the connections of those neurons near the spinal cord connect through the spinal cord to the brain, and for a long time give this illusion of a phantom limb, which you may have read. There's a marvelous public sort of popular science book called Phantoms in the Brain by V.S. Ramachandran. I encourage you to read it. So essentially, the brain thinks the limb is there because the neurons are still there next to the spinal cord, and they think they're getting sensation. Okay. After a while, there are therapies which persuade the rewiring of these circuits to happen, and phantom limb sensation can be diminished somewhat. And indeed, the area then that is supposed to receive sensation from the limb gets taken over by so no cortex sits around doing nothing. Okay, as an example, in congenitally blind people, their entire visual cortex, we devote a lot of cortex to vision because we're very visual people. The visual cortex actually gets taken over by touch sensation cortex, because blind people use their fingers, use their hands to feel the environment. So no cortex goes wasted, cortex gets taken over. And I hope I've answered your question to some extent.
I think so. So I'll go with one of the maybe simpler questions for you first. One of our attendees wanted to know a bit more about the role of support cells, where you mentioned that there are two kinds of cells. So maybe if you could share something.
So support cells provide actually factors that the neurons need to survive, to live, even to grow, and to maintain their connection. So when one neuron connects with another neuron, actually the support cell is in there forming a triumvirate of connectivity, right? So the support cells also help augment the neuron's ability to work. So support cells are not to be discounted in any way. Then the system cannot work without them. So they just have different jobs at all. That's all.
Okay. So we have another really interesting question from Samyukta in the audience. So firstly, thanks you for the lecture. And I know this is something I think which came from what you spoke about enriched environment, Shubha. So her question is there such a thing as overstimulation in human environments similar to the mouse model wherein it only causes neurogenesis and limits gliogenesis?
Thank you. Thank you. So I didn't intend to give the impression that enriched environment causes the gene to, so when I said we get only neurons and do not get support cells, that was a genetic manipulation we did. Okay, that we discovered a factor that makes more neurons instead of support cells. The enriched environment does this by signaling stem cells that are normally present in this part of the brain. We hypothesize that it may be acting through this factor. We haven't shown it yet. Okay, many factors control whether the stem cells that are present in our brain today are being, how they are being instructed to produce more neurons. Okay, attending a lecture like this, tick, 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 your neurons you're making new connections and so on in the brain. Anything new you learn creates new connections. Any form of new experiences so informs the brain to create new neurons and new connections. And we hypothesize that this may happen through factors such as the ones we have discovered. We did it by artificially forcing expression that can't be turned off. So it's a very extreme situation in which you get a sense of what your gene can do. And then you try and look for it in normal situations of enriched environment.
Okay. And just as a follow-up to that, Shubha, just in these cases where you do cause excessive neurogenesis or more neurons to be formed, have you at all ever seen anything that ties this back to any kind of behavior change or change in the mouse?
I'm sorry, I didn't understand.
So when you do through genetic manipulation, you're able to have excessive neurons, more neurons, or flip the balance rather. Does this lead to any kind of behavior change?
So we actually are doing those experiments. We have because we have to make enough of these neurons to form in order to change it. Very, very tantalizing question, but science is an ongoing process and we don't have the answer.
Okay. There's another really interesting question from V. Selva Ganapati, and I think this also came from a question that came to my mind. And his question is, how does the brain restructure as and when a child achieves major gross motor milestones, and what is the influence of the senses on the same? And I guess my question also was, in this development of the brain, how much of this is embryonic and how much happens, during early childhood, and then of course the external world playing an influence on this.
Very nice, very nice. So how much of the development happens embryonic and after birth really depends on which species you're looking at. Okay, and let's not be too arrogant about our humans being more important. I mean, if you've seen movies of, if you've seen a chick hatch, first the thing can peck itself out of its own egg, and then it falls floppity and gets a little rest. Then the minute it has done so, it is up and it's going tick 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 pecking for food. Okay, which human baby can do that? Okay, so human babies are born very, very helpless, as we all know. They need huge amounts of maternal care. I mean, newborn fish, the minute it escapes its little casing, it can start swimming around, right? So humans need a lot of care. We are born with very, very underdeveloped circuits, and the circuit development happens after birth. Okay, and naturally, experience of the world. Experience is a word that scientists use for essentially what the senses bring to it. Okay, a child needs to experience light, sound, color in order to be able to make sense of those things. right, being raised in environments that don't have regular amounts of all of these things can actually distort how the circuits get wired. So in terms of motor milestones, you can almost, so I'm not an expert in this since I have not studied human child neurobiology, but you can pretty much see it happen, right? Have you seen a child trying to clap their hands with their newborn? They'll do that, and then suddenly they will connect and once they're able to connect, they never miss again, right? You can see the circuitry being tooled. Have you seen a little infant in a pram where it will suddenly go chick and all of its arms and legs will fly out at once? Okay, over time, it learns to have a more guided, the muscles that have to contract and stretch sort of learn to work together so that everything doesn't all fly out at once. Even as a baby walks, that's motor control circuits being shaped. So every action the baby does shapes its circuitry. Every circuitry change enables more action. So you can see that happening, and you don't actually need a neuroscientist to tell you. But I hope that answered some of your questions.
No, I think that absolutely does. And I just probably want to add on a connected note. If you check out our activity handbook in the exhibition, there is a case study of Jeannie Wilder, who was a young child who they actually looked at to understand the development of language, how if you were isolated from society, what happened to the ability to be able to learn this, which I think does tie in a bit to what you were speaking, Shubha, about how you pick up a lot of things early on, and this is hugely influenced by the stimulus you receive. There's a couple of questions in the chat box also. I'm going to pull up some of those. One is, I think, about synesthesia. So Unikrishnan has asked, I've heard that there are people who can see colors where they see numbers or vice versa. How does this happen? So I think this is about synesthesia.
So synesthesia, again, actually, Ramachandran has some of the best hypotheses for this. The thinking is that connectivities between parts of the brain that process, say, numbers and colors are sort of cross-connected. And when there is a percept of a number, it also activates the percept of a color. This is the sort of hypothesis. But, to actually do the experiment, you would have to begin to start poking around the brain of a synesthete, which is not really nice to do. But this is sort of a very insightful hypothesis of Ramachandran that because connections get triggered in areas that are not present in non-synesthetes, numbers have color overtones to them. So it's all circuitry, obviously. A number doesn't have a color in it. So, but the percept of a color is triggered.
So it's mostly essentially in the mind then, not so much in the perception.
It very likely comes from connectivity. So even this thing called mind comes from circuits. The mind is not a nebulous amorphous thing. It arises out of the functioning of a circuit.
Yeah. I think for those who are interested more in this, I could also refer you to Professor Upinder Bhalla's talk, which happened last weekend, where he also spoke very much in detail about the circuits that are formed in the brain and where does the concept of the mind come from.
I love
I'm going to take another question, which is sort of interesting. I'm going to elaborate upon it a bit. So Bhavi's question is that not all cells that are born, live forever, right? So some of these cells are going to die. What happens to the circuitry then? So I guess this really comes to, are these circuits the exact same neurons that are always maintained through our brain? Are they changing?
And, very nice, very nice. So this is a very cool question. In different parts of the nervous system, different things happen. Very oddly, motor neurons that reside in the spinal cord that normally connect to muscle, and literally when you contract muscle and say biceps, you're doing the signaling. Very oddly, motor neurons are born in way large numbers than needed in the embryo. And when they start, they're born and they sit in the spinal cord, and then they grow out to the limbs. Those that connect to target muscle get from that target muscle a survival factor and survive. And those that don't die. It is unclear, and this highly somewhat wasteful process. Is unclear why it happens, but it happens this way, that neurons are dependent on targets for survival cues. Now, that's a developmental phenomenon. I'll give another example of a completely different system, olfaction. Okay, the neurons in our nasal epithelium way inside, the ones that stick goes and scrapes when you do a COVID test, those neurons are actually exposed to the elements, right? I mean, they're covered in a protective layer of mucus, but they're pretty much exposed to the elements. They have a hard life and they die, okay, routinely. And their connections to the brain, where we get into our olfactory bulb, where our sense of smell gets started, their connections to the brain will naturally also wither away. But fear not, there are stem cells in the nasal epithelium which make new neurons, which magically follow the same route they're supposed to and make those same connections. So broadly over your life, your sense of smell is broadly maintained, but it is susceptible to change, in that if some dramatic perturbation could happen where things don't exactly smell the way they used to. And that's partly because your neurons continue to regenerate and continue to connect to the brain. So it's an ongoing shifting situation.
Okay. There's another question. So there are lots of questions, Shubha, so I'm going to just like slightly throw them at you. There's another question, which is from Subbu, and he of course thanks you for a fantastic talk. And he wants to know whether through the use of technology we are able to say, do we get a sense of how good or bad our state of our sensory perception is, and how, in other words, how evolved or otherwise. I think it might also be an interesting thing for us to know from you if our sensory perception abilities have evolved over time, not just say when we came from apes to humans, but even within, do we see that as something that could happen going forward as well? Or do you have any speculative ideas of where we might?
Very cool. I have two answers. One, are there technologies that get a picture of how good or bad your state of sensory perception is? Yeah. Have you ever been to a doctor and had an auditory test? They'll hold a tuning fork, or they'll now put headphones, and then they will make you hear frequencies that go to some high pitch that you'll stop hearing. And with age, it's very predictable that your neurons in your cochlea are pretty much dead and you can't hear those high pitches anymore. So yeah, there are technologies available to do all of this. Sensory perception, you can do it yourself with a good friend. Have the friend take 2 toothpicks and close your eyes. And the simple question is, are there 2 or 1? Okay, the person can also poke twice. At what distance of closeness will you be able to discern that there are 2 pokes, not 1? Okay, out here, in front of your fingertips, very close, you'll be able to say there are 2. Okay, out here, barely difficult. Okay, and that connects to that picture of the human I showed you with a very disproportionate representation in the brain, where you have a lot of brain space devoted to a small area. Your resolution is very, very high. You don't need technology, you need toothpicks. Okay, and in parts that are less well represented, you won't be able to tell. So that, what was the second part of your question?
I guess, what does evolution, how
evolutionarily, well, we, during our lifetimes, okay, our sensory circuit is plastic. It's changeable. A pianist uses fingers. Anything you use will begin to occupy more territory and become richer in its. So the more you use a modality, the more brain power, brain space, and circuitry gets developed. So during the course of your life, you are evolving. We are all evolving. And the mantra there is use it or lose it. So people with children do introduce them to a multiple different experiences, so that they may choose which ones they want to grow in. And the more you practice at something, the better you become at it. Look at dancers. You can tell, okay, the same performance, the same piece. Take the very basic beginning Bharatanatyam piece, Allaripu, okay? If a trained dancer does it versus somebody who's learned for 2 years, you can tell the difference. So even though you may not be a dancer, okay, your eyes can pick up the small micro differences in how the body is held and how the curvature, the command of the control of the muscles that give you that command and stature. An untrained eye can tell A is different from B, right? Because there is evolution of these circuits and timing and precision. When a swimmer trains to shave those microseconds off of their record, what are they doing? They're training their circuits to kick in time such that a precision is achieved. Right? So that's what's happening.
So I'm just going to, kind of jump the queue and ask a question that came up while you were explaining this, Shubha. And this is whether these, these things that, these expansions that are caused by practice or by, doing things again and again, are these heritable?
I'm sorry. Can you repeat the question?
So the changes that you said just now that happen, due to doing practice or, developing a skill or, fine tuning a sense of perception over your lifetime. There's a question that came in while you were speaking, asking whether these changes are heritable or they can be inherited from one generation.
The short answer is no. You have to train yourself, each individual. But the deeper answer is the potential to have some circuit hum a little more easily than another. That potential is heritable because after all, we are the product of our genes. If some circuit is more amenable to being trained up because of the combination of genes that controlled its initial growth, that is heritable. Okay, but it's very difficult to separate heredity from environment. A child of excellent singers is going to grow in an environment of excellent music. The early experiences are going to be excellent music. So to sort out whether the child's genetic circuitry was predisposed in the musical space or the experiences made it so, right? That's harder to sort out, but both things play a role. It's not necessary that a child of great musicians will develop music ability. It's not necessary at all, right? But given all other things being equal, if you're exposed to advanced music from childhood, the chances are you're going to get, okay, let me put it this way. Genes can define which strata you can belong to, right? I, for example, am never going to become an Olympic swimmer. So if the swimming strata is like this, right? I'm probably there, maybe there, right? But if I had practiced swimming from childhood, heck, if even I practice it now, I can get as high up within my strata or even maybe cross a strata. Okay, so this heredity can set up some broad ballpark, and these strata can be overlapping too. It's a heredity can set up broad ballparks, and after that, it's practice and experience. Some things are just limited, right? Your height is, again, limited by your genes. You can work at it and get it higher or not. But after all, look at the average Indian height and look at the average height of, I don't know, some North European type. If there's like a whole foot difference in average height. A lot of it is nutrition, a lot of it is exercise, but a lot of it is also genes. Did that answer?
Absolutely. I'm just checking how much time we have. Okay, we have a few more minutes. I could ask you a couple of questions. So one question that came in a couple of times is from S. Gopalakrishnan. And I think the question is, about reality, right? You began with this, your talk with how you said that, our ability to sense and perceive the world and know or to say that we know the world around us is what helps us construct the sense of reality. So in cases of mental illnesses or such as schizophrenia, where you might then lose touch or you might experience a kind of different sort of reality, do you have any insights to share as to what might happen in the brain or any other thoughts on this?
I'm going to take this question to a very philosophical space. How can we know anybody else's reality? Okay, forget schizophrenics or anybody. How can I know what your reality is in your head? How can I try and explain to me what red is? Okay, how do I know your red is the same as my red? the best we can do is reference it with, well, it's a color that kind of lies, more on the orangey side, if it's not on the yellow side of orange, but on the more brighter, darker side. Try and convey what red is. It's impossible, right? Because your percept of redness is because of the way the photoreceptors in your eye fire, and then you develop that percept, which we all call red. Okay. If one shows a bunch of colored blocks, most of us will identify a particular block as red. But the percept of redness, which maps to the percept of reality. Who can communicate with that, right? So that's sort of the philosophical answer to that.
Okay, there are, I'll take last two questions and then we will wrap up. So the first question is sort of, is there a peak for our sensory perception, right? Our ability to sense things and perceive things, does this get worse, better over the course of our lifetime? And does this differ for different senses? So is there a peak in our life when this is best? And yeah, maybe you could answer that and then I'll come to the next.
Well, yes, of course, everything ages, but see, where is this question coming from and why is it, why do you want to know, right? In the abstract, yes, of course, everything ages. I just gave the illustration of sound. You can't hear things out, right? But to put a practical twist on it, what one brings from the experience of one's lived events in life. Okay, if you've spent 40 years listening to fantastic classical music, at the end of the day, you're going to appreciate music, perhaps with a fine nuance at the age of 45, even though if you do a sensory test, your cochlea is not going to be able to hear some. Sensation is just sensation, right? It's what your brain makes of it finally that makes our lives rich. So depending on where you came from with the question, yeah.
So maybe Samita, if you'd like to add further detail to your question, you can add it in and we can check with Shubha. In the meanwhile, I think we can take one last question. So this is in terms of the plasticity of the brain in Shubha, are you able
to go ahead? I'm here. Go ahead.
Okay. So this is a question about the plasticity of the brain of an adult versus a child. And are there any major differences that are there and what this might lead to?
I have to ask whoever asked this question, have you interacted with children, taught them stuff, and have you seen a child recover from a gaping gash in their arm, which one day is gaping and bleeding, and 2 days later, it's like, nothing happened, no, nothing. Okay, I mean, the young system just does everything faster, everything. Forget child. The teenage brain is possibly amongst the most plastic. Everything is, anybody who's lived with a teenager, every day is different. Every day is like a new species almost. It's a time of great plasticity and a time of changing thoughts, changing goals, changing decisions. Much can be explored, much can be learned. So yeah, things actually go through so different systems kick in their peak activity at different times. There's a whole growth phase in terms of the brain. The teenage brain goes through its expansive plasticity. And then after that, it is refining and sort of adding experience and maturity in more ways than one to the circuits, the connections, their performance.
So I think Samyukta also got back to us about her question, and she says that she thought about it in the context of taste, as in what is the best age to say taste a great meal. And she says that your explanation, she understood your explanation, and that in the sense that if you has refined her palate over time, she'd probably appreciate it.
Brilliant. Better. Yes, it's perfect. It's a perfect explanation. It's a perfect explanation. And to map it to my explanation of dance, the teenage brain is plastic and can probably learn new steps and entire new compositions quickly. But to look at an experienced dancer at an older age is a treat. Okay, so every part of life gives you something.
Okay, I think this was a very good time for us to begin wrapping up. We've had so many questions. I'm so glad that everyone was so engaged and had a lot of things to ask, Shubha. Thank you so much. I think we've all had a very insightful evening today. And I think if you have enjoyed this lecture, please do sign up for a live lab walkthrough with Anzal from Professor Upinder Palla's lab, who's going to be showing you how some of these imaging and some of these experiments that even Shubha talked about, how they are actually conducted. So you could join in for that. And if you like this, do also check out SP Arun's lecture, The Incredible Perception Lab Inside Your Head. He also talks about some very fascinating experiments you can do to begin deciphering how your sensory system works. I'd also like to suggest 2 connected exhibits in our exhibition season. One, which is called Playing with Reality by Anagram, and it comes back to the sort of philosophy point at the point that Shubha brought up, right? How do you know what someone else's reality is, and how do you begin to understand that? So there's this exhibit, please do check it out. And you can, and for those who are interested in schizophrenia, you can also see the exhibit Schizophrenia and the Brain, which is by a team from NIMHANS. Professor Urvaksh Mehta and Dr. Makuran Pantoji, which talks about the some of the biological underpinnings of this condition and the research that's currently being done to understand this. I'd also like to encourage everyone in the audience to please share your thoughts in our feedback form, register for our upcoming programs, and check out the exhibition website. It's been absolutely fascinating having you here today, Shubha, this Sunday evening, and I think it was a really live and interactive session with our audience. So thank you so much, everyone, and Shubha for being here today evening, and we look forward to seeing you for the next till the end of the season. Thank you, everyone.
I'm so very glad. Thank you so much.